Okay, so welcome to this next video in which we are discussing intestinal stem cells. Okay, so we now want to discuss what it is that maintains intestinal stem cells as intestinal stem cells. Okay, and we've discussed that the reason that there is this specific niche where if a daughter cell remains in that niche, it remains a stem cell, and if it gets pushed out, it becomes a terminally differentiated cell. The reason for that is because the pan of cells are secreting Wnt ligands, and these Wnt ligands are going to set up a signaling cascade within the cells that maintains them as stem cells, basically. Okay, so we want to see this pathway known as the Wnt beta-catenin pathway and how it's going to change gene expression within these cells to maintain them as stem cells. Okay, right, so we're going to start off by discussing the receptors for Wnt ligands. Okay, so there are actually two proteins which are going to be involved in binding to the Wnt ligands in the Wnt beta-catenin pathway. Okay, and these are going to be Frizzled receptors along with LRP5 or LRP6. Okay, so we'll start off by discussing the Frizzled receptors, and then we'll move on to the LRP5 and the LRP6. Okay, so Frizzled receptors. Now there is not just one Frizzled receptor, there are multiple Frizzled receptors, okay? And they are all Gprotein-coupled receptors. So they are contained within the bigger family of receptors known as the GPCRs, okay? So this is a symbol from maths, which means contained within, okay? So the set of all Frizzled receptors is contained within the set of all Gprotein-coupled receptors. Okay, right, so I suppose I should probably write out G-protein-coupled receptors at least once in this video. Okay, so G-protein and then coupled, that's the C, and then the R is for receptors. Okay, right, so what I want to start off with is a basic discussion of the different domains of a G-protein-coupled receptor. Then I want to discuss the different families of G-protein-coupled receptors. I want to see which family of G-protein-coupled receptors the Frizzled receptors fit into, and then we'll discuss the diversity of Frizzled receptors, and then we'll move on to the LRP5 and LRP6 proteins. Okay, right, so G-protein-coupled receptors then. Okay, let these two lines here represent the outer leaflet of phospholipids and the inner leaflets of phospholipids. Okay, so the outer and inner layer of phospholipids that make up the phospholipid bilayer. Okay, now all G-protein-coupled receptors have a shared membrane-spanning topology. Okay, now in humans there are over 800 G-protein-coupled receptors, and they all have the same membrane-spanning topology that I'm about to show you. Okay, so they start with their amino terminus extracellularly. Okay, so actually the first thing to say is that they are all single polypeptides, so they're not complexes of multiple polypeptides, they are a single polypeptide. Here's the amino terminus, then this line represents the polymer of amino acids. Okay, it spans the membrane, so it goes from the outside into the inside, then it will span the membrane again a third time, a fourth time, a fifth time, a sixth time, and then a seventh time, and then you'll have the carboxylic acid terminus intracellularly. Okay, so these membrane spanning portions, which I will colour in red here, these are membrane spanning alpha helices. Okay, so all of these are membrane spanning alpha helices. So even though I've shown them as straight lines, in actual fact they are alpha helices. Okay, so they will have a spring-like structure. So the polypeptide will spiral round in a spring like so. Okay, and that sort of a structure is known as an alpha helix. And the polypeptide within this membrane-spanning alpha helix will be mainly made up of hydrophobic residues, and these hydrophobic residues will stick into the hydrophobic core of the phospholipid bilayer, and those will help to stabilize this membrane-spanning alpha helix within the hydrophobic core of the phospholipid bilayer. Okay, so I won't actually draw them as springs or alpha helices, but they are in fact alpha helices. Okay, so these 7 membrane-spanning alpha helices, they all have names. Okay, so the first membrane-spanning alpha helix is called TM1, standing for transmembrane alpha helix 1. Okay, so this TM is for transmembrane. Trans means across, and then membrane, so it's across the membrane. Okay, then the second membrane-spanning alpha helix here is known as TM2, the third membrane-spanning alpha helix is known as TM3, the fourth membrane-spanning alpha helix is known as TM4, the fifth membrane-spanning alpha helix is TM5, the sixth is TM6, and then the seventh, which I'll also label up, is known as TM7. Okay, so there's TM1 through TM7. Okay, now there are more domains that are conserved that have names other than just the 7 membrane-spanning alpha helices. And by the way, I'd just like to mention that you find this structure within all 800 of the G-protein-coupled receptors within humans. And you also find G-protein-coupled receptors outside of humans as well. You know, I think the total number of G-protein-coupled receptors that biologists have so far found in living organisms stacks up to around 30,000 now, and they all have this same 7 transmembrane domain structure. Okay, and it is not really well understood why nature has run with this domain structure at all. Okay, we don't know what is so significant about the 7 membrane-spanning alpha helices. Why has nature made such great use of it? What was the major success that this had? Okay, but anyway, it's there. So let's continue with the different domains of these G-protein coupled receptors. So other labeled domains are these 3 loops that are on the intracellular side here that are now coloured in orange. Okay, so these 3 loops here, these are known as the intracellular loops. Okay, so intra means within, cellular means cell, so within the cell, and these are loops that are within the cell. Okay, now for short, the intracellular loops are abbreviated to the ICLs. I for intra, C for cellular, L for loop. Okay, so the ICLs. Now the first intracellular loop, which is between TM1 and TM2, that will be called ICL1. Okay, so this one here, this is ICL1. The second intracellular loop between TM3 and TM4, that will be called ICL2 here. Okay, and the third intracellular loop between TM5 and TM6, that will be called ICL3. Okay, so those are the 3 intracellular loops of a G-protein coupled receptor. Then on the extracellular side, we also have extracellular loops, which I'll highlight in turquoise here. So these 3 loops that are on the extracellular side of the G-protein coupled receptor, these are the extracellular loops, and extracellular loop is usually abbreviated to ECL, E for extra, C for cellular, L for loop. So again, you have 3 of these things. So there is extracellular loop 1, ECL1, which is this one between TM2 and TM3, the ECL2, which is between TM4 and TM5, and then the ECL3, which is between TM6 and TM7. Okay, right, so that's the intracellular loops, the ICLs, and the extracellular loops, the ECLs. Okay, additional structures that these G-protein coupled receptors all share is that they share this portion that is prior to the TM1 domain. Okay, and this is known as the amino-terminal domain, or the ATD for short. So ATD, I'll write it down here, stands for the A is for amino, the T is then for terminal, okay, and then the D is for domain. So this is the domain of the protein, which is close to the amino terminus of the protein. Okay, so that's the ATD. Now some people will even call the amino terminal domain the ATED. Okay, so this is another bit of nomenclature you might see. Okay, so what does this stand for? Well, the A once again stands for amino. The T stands for terminal, but then the ED here stands for ectodomain. Okay, now ecto just means on the outside, so this just means the domain that is on the outside of the cell. Okay, finally, you then have this portion that's after TM7, and this is known as the C-terminal domain, okay, or CTD for short. So this is the portion that is near the C-terminus of the polypeptide. Okay, so that's the general domain structure of all G-protein coupled receptors, not just the Frizzled receptors, that's all of them there. Okay, right. Now what I want to do is discuss the different families of G-protein coupled receptors, because there are over 800 G-protein coupled receptors in humans alone. Okay, so to give us some sort of handling of these over 800 different receptors, we group them into families. So I'd like to discuss the grouping of G-protein coupled receptors into families. And I'm doing this so that we get a big overall picture of where the Frizzled receptors fit into the family of G-protein coupled receptors. Okay, they are actually in the 5th family, so we're going to have to go through all 5 families to get to the final family. Okay, but it will be worth it. It will give us a good understanding of where the Frizzled receptors fit into this massive, great superfamily of G-protein coupled receptors. Okay, so we'll start with family number 1 of G-protein coupled receptors. Now this first family of G-protein coupled receptors is easily the most important family of G-protein coupled receptors. It is a massive, great family of G-protein coupled receptors, which contains most examples, basically. So we said that there are over 800 G-protein coupled receptors within humans. Of these around 800, 750 or thereabouts are within this rhodopsin family of G-protein coupled receptors. So it is an enormous, great family. Okay, so what characteristic feature does a G-protein coupled receptor have to be in this rhodopsin family of G-protein coupled receptors? Well, once again, I'll draw the cell membrane here. Okay, here is the G-protein coupled receptor with the conserved domains that we've talked about here. So here's the amino terminus, and here's the carboxylic acid terminus. Okay, and the characteristic feature that you have to have in order to be a rhodopsin family G-protein coupled receptor is that you have to bind your ligand in the extracellular 1/3 of the membrane-spanning alpha helices. Okay, so we have these 7 membrane-spanning alpha helices here in red. Okay, the ligand for a rhodopsin family G-protein coupled receptor is going to bind to residues that are in the outer 1/3 of the 7 membrane-spanning alpha helices. Okay, so here is the outer 1/3, okay, of the 7 membrane-spanning alpha helices, and that purple blob there is meant to represent our ligand binding to residues within those outer thirds of those 7 membrane-spanning alpha helices. Now, on my picture, I've specifically drawn it interacting with the 3rd, the 4th, and the 5th membrane-spanning alpha helices. But in reality, it can interact with many different ones in different examples of rhodopsin family G-protein coupled receptors. Okay, so the characteristic feature then of rhodopsin family G-protein coupled receptors is that they bind their ligand to the outer 1/3 of the membrane-spanning alpha helices then. Okay, right. They have quite small amino terminal domains as well, okay, because the amino terminal domain isn't going to be involved in the binding of the ligand to the receptor in this case. Okay, so examples of G-protein coupled receptors that are within this family. So the rhodopsin receptor itself, which is the receptor after which the entire family is named. This is a rhodopsin family G-protein coupled receptor. It is the receptor in the rod cells of the retina that responds to light. Okay, it's an incredibly well-studied G-protein coupled receptor, and that's why this whole family is named after it. Now you might say, well, what's the ligand in that case? Okay, because surely if it's activated by the light, what's the ligand? Well, usually rhodopsin receptors have bound to them a molecule that is stopping them from becoming active, and this molecule is 11-cis retinal. Okay, and this is always bound to the rhodopsin receptors in the dark, okay, and it's stopping the rhodopsin receptors from being active. Now, when light hits 11-cis retinal, it causes photoisomerization, and it converts the 11-cis retinal into all trans retinal. Okay, so the double bond in this structure that was off the 11th carbon goes from being in the cis conformation to being in the trans conformation, and that we then get all trans retinal. This then cleaves off the rhodopsin receptor, okay, which is often on its own known as the opsin receptor, okay, and then that is capable of then interacting with a heterotrimeric G protein known as transducin. Okay, so that's an example of a rhodopsin family G protein coupled receptor. Other famous examples are, for instance, the adrenoreceptors. Okay, so all of the receptors for adrenaline and noradrenaline, they are rhodopsin family G protein coupled receptors. The muscarinic acetylcholine receptors are also rhodopsin family G protein coupled receptors. The histamine receptors, okay, the serotonin receptors, they are all rhodopsin family G protein coupled receptors, at least most of the serotonin receptors. The 5-HT3 receptors are actually not G protein coupled receptors at all, but rather are ligand-gated ion channels. Okay, but all the other serotonin receptors apart from those are rhodopsin family G protein coupled receptors. Okay, so that's the rhodopsin family of G protein coupled receptors then. Let's move on to the family number 2. Okay, so family number 2 of G protein coupled receptors is what's known as the secretin family of G protein coupled receptors. Okay, so let's underline this in blue. So, this secretin family of G protein coupled receptors, this is much smaller than the rhodopsin family of G protein coupled receptors, but all of the other families of G protein coupled receptors are dwarfed by the rhodopsin family of G protein coupled receptors. Okay, so what special feature do you have to have in order to be a secretin family G protein coupled receptor? Well, these G protein coupled receptors are generally going to have very large amino terminal domains, okay, so much larger than the rhodopsin family G protein coupled receptors. Okay, so here's the amino terminus, here is the carboxylic acid terminus, and they are generally going to have ligands that are peptides. Okay, so whereas the rhodopsin family G protein coupled receptors, they can have very small molecules as their ligands. Rhodopsin has this little molecule 11-cis retinal. Adrenoreceptors have adrenaline and noradrenaline, small molecules. Okay, muscarinic acetylcholine receptors, acetylcholine, a small molecule. 5-HT, serotonin, is a small molecule. Okay, these receptors in the secretin family, they nearly all have polypeptides as their ligands. Okay, and more importantly, the actual characteristic feature of these secretin family G protein coupled receptors is where they bind their ligand. Basically, they bind their ligand in a little slot between the amino terminal domain, which is here, which I'll colour in orange here, so this is the amino terminal domain, and the transmembrane domain, which is underneath. So the domain consisting of these 7 membrane-spanning alpha helices here, this is the transmembrane domain. So basically, the ligand shown here in blue, this is going to bind in this slot between the amino terminal domain above and the transmembrane domain below. Okay, right. What example G-protein coupled receptors are within this secretin family of G-protein coupled receptors? Well, all of the receptors for calcitonin are secretin family G-protein coupled receptors. The receptors for parathyroid hormone, okay, again another peptide hormone, they are all secretin family G-protein coupled receptors. Now, calcitonin and parathyroid hormone, which is also known as PTH, those are both involved in the regulation of calcium concentration within the blood. Okay, another potentially more famous example are the receptors for glucagon, which are on the liver. Okay, so glucagon is very important in control of blood glucose level. Okay, insulin takes blood glucose down, glucagon brings it back up, and the receptors for glucagon are mainly centered on the liver, and they are secretin family G-protein coupled receptors. Okay, right, so that's the second family of G-protein coupled receptors. Let's now move on to the third family of G-protein coupled receptors. So the third family of G-protein coupled receptors are known as the glutamate family of G-protein coupled receptors. And you might be able to guess from the name what receptors are going to be within this glutamate family of G-protein coupled receptors. Okay, right, so let's start with what characteristic feature you have to have in order to count as a glutamate family G-protein coupled receptor. So once again, I'll draw the cell membrane here. Okay, the outer leaflet and the inner leaflet. Now, once again, you have your amino terminus on the extracellular side. Okay, then you're going to have a special domain within your amino terminal domain. Okay, like so. Then you've got your 7 membrane-spanning alpha helices here, and then here is the carboxylic acid terminus of our polypeptide down here. Okay, so this special domain here, which I'm now circling in turquoise, this is what's known as the venous flytrap domain. Okay, so this is called a venous flytrap domain. Now, the reason it's called this is that it's analogous to a venous flytrap. So this venous flytrap domain is going to bind the ligand. So the ligand is going to bind here. And the idea is that the ligand will bind between those two halves of the venous flytrap domain. And when the ligand binds, those two halves will close around the ligand and trap it in there. So it's analogous to a venous flytrap closing around a fly which has just landed in it. Okay, right, so that's the characteristic feature that all glutamate family G-protein coupled receptors have. They have a venous flytrap domain which is going to close around the ligand molecule once it binds there. Okay, right, so what receptors are within the glutamate family of G-protein coupled receptors? Well, clearly there are going to be some receptors for glutamate. Specifically, the family of glutamate receptors which are within this glutamate family G-protein coupled receptors are the metabotropic glutamate receptors. Okay, so glutamate is the most important excitatory neurotransmitter within the brain. Okay, however, the most important receptors for glutamate are not G-protein coupled receptors. They are ligand-gated ion channels. The ionotropic glutamate receptors are the main receptors, and as I say, they're ligand-gated ion channels. However, there is another family of glutamate receptors, which is known as the metabotropic glutamate receptors, or for short, the M is for metabotropic, the GLU is for glutamate, and then the R is for receptors, so M-GLU-Rs. Okay, now there are 8 metabotropic glutamate receptors, and they are all receptors for glutamate that have one of these venous flytrap domains within their amino terminal domain, which binds to the glutamate. Okay, another family of receptors that is within this glutamate family of G-protein coupled receptors are the GABA B receptors. Now, glutamate is the most important excitatory neurotransmitter within the brain. GABA, or gamma-aminobutyric acid, is the most important inhibitory neurotransmitter within the brain. Okay, now again, the most important receptors for GABA are not G-protein-coupled receptors. They are again ligand-gated ion channels, and that family of receptors is known as the GABA-A receptors rather than GABA-B receptors. However, there are some receptors which are G-protein-coupled receptors and which are receptors for GABA, and these are called the GABA-B receptors. Okay, and these are also glutamate family G-protein coupled receptors. So I find it kind of interesting that these two sort of side receptors for the two most important neurotransmitters within the brain, the most important excitatory one, glutamate, and the most important inhibitory one, GABA, are both within this family of glutamate family G-protein coupled receptors. Okay, right, so that's that family of G-protein coupled receptors. Now, let's go on to the fourth family. Okay, so over the page. Right, so the fourth family then of G-protein coupled receptors is the family that no one can ever remember. Okay, this is the family of adhesion family G-protein coupled receptors. Okay, right. Now, the characteristic feature that all adhesion family G-protein coupled receptors share is that they all have an extremely large amino terminal domain. Okay, so let's draw this here. So we have a very, very big amino terminal domain here. Okay, then we have the 7 membrane spanning alpha helices, which we can't get out of. You have to have those, otherwise you're not a G-protein coupled receptor. Then you have your carboxylic acid terminus down here. Okay, now adhesion family G-protein coupled receptors do not bind to your conventional idea of a ligand. Okay, they do not bind to a free molecule that's floating around in the extracellular fluid. Instead, they bind to components of the extracellular matrix. Okay, so they use this very large amino terminal domain you have here, okay, to bind to components of the extracellular matrix. Okay, so this is representing a component of the ECM, the extracellular matrix here. Okay, so we'll colour this in red. Okay, so adhesion family G-protein coupled receptors then bind to a component of the extracellular matrix, the ECM. Okay, and that's all we're going to say about adhesion family G-protein coupled receptors. Now we come on to the fifth and final family, which is the star of the show, basically. This is what we've been building up to. This is the family that contains the Frizzled receptors. Okay, now the Frizzled receptors are quite an important member of this family, so the whole family is named after them. Okay, so this is called the Frizzled/Taste2 family. Okay, named after the two most important members of this family, which are the Frizzled receptors and also the Taste2 receptors. Okay, right. So it's fair to say that everything that comes into this family is just everything that doesn't fit into any other family, basically. Okay, so this family of G-protein coupled receptors, they have a more normal type of ligand than the adhesion family G-protein coupled receptors, and they bind to their ligand using their amino terminal domain. Okay, so we'll draw one here. Here's the amino terminus of this G-protein coupled receptor, here's the carboxylic acid terminus, and basically the ligand molecule is going to bind to the amino terminal domain of this G-protein coupled receptor, like so. So here is the ligand molecule in purple here. Okay, now it basically binds to the amino terminal domain, but you don't have any of the fancy things that we had before. Okay, so firstly, it's not an adhesion family G-protein coupled receptor because we're binding to a free ligand, not a component of the extracellular matrix. It's not going to be a glutamate family G-protein coupled receptor because we're not going to have a venous flytrap domain. We're not going to be a secretin family G-protein coupled receptor because we're not going to bind our ligand in this slot between the amino terminal domain and the transmembrane domain. And finally, we're not going to be a rhodopsin family G-protein coupled receptor because we're binding our ligand in the amino terminal domain, not the transmembrane domain. Okay, so everything else where you just bind a ligand to the amino terminal domain, that's grouped into this Frizzled/taste2 receptor family. Okay, and the members of this family, the main members of this family, are the Frizzled receptors, which we need to talk now about. Okay, and then also the taste2 receptors, which are involved in sensing bitter tasting molecules in the gustatory system. Okay, so the Frizzled receptors then, finally. So we've discussed all of those families so that you have a broad picture of G-protein-coupled receptors and you understand where the Frizzled receptors fit into this family of G-protein-coupled receptors. Okay, so there are 10 different Frizzled receptors. Okay, and by the way, Frizzled receptors are often abbreviated either to FZ, okay, so that's an abbreviation for Frizzled receptors, or some people abbreviate them to FZD. It doesn't really matter. I think I will use just FZ, okay, but you might see FZD, but it is just another abbreviation for Frizzled. Okay, now there are 10 different Frizzled receptors, and the naming is perfect, okay? So they are just named Frizzled 1, Frizzled 2, Frizzled 3, all the way up to Frizzled 10 receptor. Okay, and those are the 10 different types of Frizzled receptor. Okay, right. So these are one of the receptors that is going to be involved in sensing the presence of extracellular Wnt ligands. Okay, but they are not the only one. There's going to be another one that's involved as well. And I now want to discuss this other receptor that's involved in binding to Wnt. Now, the Frizzled receptor gets the better title. The Frizzled receptors are called the receptors for Wnt. Okay, these other receptors are known as the co-receptors for Wnt. Okay, so these are the ones which get the lesser title, if you like, but they really have as much of a function in sensing the Wnt as the Frizzled receptors. Okay, but that's the way it's been called, so that's the way we'll stick to it. So these are the receptors and these are the co-receptors. Okay, right. So again, all of the Frizzled receptors, as far as we're going to be concerned, do exactly the same thing. So we're not going to distinguish between any of the single Frizzled receptors. We will just denote our Frizzled receptor as a Frizzled receptor, and it means one of these 10 Frizzled receptors, basically. Okay, right. For the co-receptors, there are just two different proteins. There is the LRP5 protein, and there is also the LRP6 protein. Okay, so we will often just write LRP5/6 to mean one of LRP5 or LRP6. Okay, but they are two separate proteins. It's not just one protein. It's just they're going to perform the same function as far as we are concerned with the Wnt beta-catenin pathway. Okay, right. So what does this stand for? What does LRP stand for? Well, it stands for low-density lipoprotein, LDL. And then the R is for receptor-related. Okay, so that just gets one R rather than two R's. Okay, because you see the dash as meaning this is one word almost. And then the P is for protein. So this is the LRP protein. Okay, and we have the LDL receptor-related protein 5, and then we also have the LDL receptor-related protein 6. So I think I should at least once write out what LDL stands for. It stands for low-density lipoprotein, and you might think, oh goodness, what's that going to have to do with all of this? Okay, well, basically, these proteins, LRP5 and LRP6, they have a very similar structure to the receptor for low-density lipoprotein that you find, for instance, on liver cells. Okay, so that's why they are called the LDL receptor-related proteins. Okay, that's the origin of their names. Okay, so let's have a look at the structure of these LRP5 and LRP6 proteins. So, once again, let us show the cell membrane here. Now again, they are single polypeptides, so we are going to draw one of them, okay? Now, both of them, as far as we're concerned, have pretty much the same domain structure, okay? So this picture will represent either LRP5 or LRP6. Okay, so here's the amino terminus of the polypeptide here. Then there are 4 in tandem, very important domains that you have here. In tandem just means one following the other, basically, in sequence. Okay, so I'm going to draw these domains as hexagons. Okay, and I'll tell you the names of these domains. They're called beta propeller domains. Okay, so I'll put this here. So these are called beta propeller domains. And basically, the reason they're called that is that they resemble propellers, so those things which drive ships forward. They resemble those. Specifically, they resemble 6-bladed propellers. Okay, so they resemble a propeller which has 6 separate blades. Okay, like so, which is why I've drawn them as hexagons. Okay, now, what are these 6 blades of a beta propeller made from? Well, they're made from beta sheets. So each one of the blades is made from a beta sheet, a small little beta sheet. So that's why it's called a beta propeller domain. Okay, then the polypeptide spans the membrane once and then finishes with its carboxylic acid terminus intracellularly. Okay, so let's colour in these beta propeller domains in blue here. Okay, right. So these 4 beta propeller domains are all given names. Okay, so the first beta propeller domain is known as P1 for propeller 1 domain. The second beta propeller domain is known as P2 for beta propeller 2 domain. Then P3, and then finally P4 for beta propeller domain 4. Okay, so that then is the structure of a basic LRP5 or an LRP6 protein. Now, so we've now discussed the receptor and the co-receptor for Wnt. Okay, now before Wnt is present within the extracellular fluid, the 2 proteins are not bound together. So the Frizzled receptor and the LRP5/LRP6 protein will not be bound together before the Wnt has arrived. The Wnt protein is going to cause the dimerization of the 2 receptors. So let's now show this. Right, so the first thing I should say is that the Wnt molecule is going to bind to the amino terminal domain of our Frizzled receptor. So we might as well start by showing that. So here we'll have the cell membrane. Okay, here is the amino terminal domain of our Frizzled receptor here. Then here is the transmembrane domain with the 7 membranes spanning alpha helices here. And then here is the carboxylic acid terminus. Okay, now we're going to have our Wnt molecule bound here. So this is a Wnt molecule here. And remember, when I say Wnt, I mean it's one of those 19 Wnt molecules that we've discussed are present within mammals. Okay, so here in purple, this is our Wnt molecule. And we've got it bound to the amino terminal domain of our Frizzled receptor here, which is shown in red. Okay, so this is the amino terminal domain. Okay, right. Now the Wnt molecule is also going to bind to the LRP5 or the LRP6 protein. Now, this is why, you remember when I said, when we were discussing the 19 different Wnt proteins, okay, this is why I said there was something slightly important about this. Different Wnt molecules bind to the LRP5/6 proteins in different places. Okay, so there are 2 Wnt binding sites, and depending which type of Wnt we are using here, it will bind at a different site. So, the 2 Wnt binding sites are always between 2 beta propellers. So one of the Wnt binding sites is between the P3 and P4 propellers. Okay, so this is known as the P3-P4 binding site here, which is between those 2 beta propellers there. Okay, then there is another Wnt binding site which is between P1 and P2. And some Wnt molecules will bind at the one between P1 and P2, and some will bind at the one between P3 and P4. Okay, so each of the Wnt molecules bind to a portion of one beta propeller and then the other portion of the other beta propeller. Okay, so just because it will be easier for me to draw the one where it's bound to the interface between the P3 and the P4 beta propellers, let's say that this Wnt molecule is going to bind to the P3-P4 binding site. Okay, so let's draw this here. So here is a beta propeller. Here, this is the P3 one, and here is the P4 beta propeller here. Then we have our membrane spanning alpha helix, and then our C-terminus intracellularly like so. And then we have a beta propeller here, which is the P2 beta propeller, and then the P1 beta propeller up here, and then finally the amino terminus up there. Okay, so let's now colour in these beta propellers in blue here. So there's P1, here then is P2, then we have the P3 beta propeller here, and then the P4 beta propeller here. And the Wnt molecule has bound at this interface between the P3 and the P4 beta propellers, and that's the P3, P4 Wnt binding site. Okay, so when Wnt is present then, you get this dimerization of the Frizzled receptor here, so I'll denote this as FZ for Frizzled receptor, and then this is either LRP5 slash, or it's NRP6. Okay, right, so we will now call it there for this video, and in the next video what we'll do is discuss what the dimerized Frizzled receptor and LRP5/6 protein are now going to do.